Okay. Okay, good night, everyone. Welcome to the webinar from our PhD and master program here in ESALQ USP. And tonight we have the pleasure, the honor, the privilege to have Professor Samantha Ying with us. I will do a very short presentation. Professor Samantha Ying has a bachelor's degree on both microbiology and physical geography. He has a PhD in soil biogeochemistry, environmental and earth sciences, system sciences, and she is an assistant professor of soil biogeochemistry at the Department of Environmental Sciences in the University of California, Riverside. Me and Sam have a collaboration going on for a few years. She had received some of our students, Lucas Sartor, Hermano, and recently she got a grant from Fulbright to visit us here in Brazil in the probably, let's see how the pandemic goes, but probably in 21 or 22. So we are very excited to have her here very soon, Sam, in Brazil. And also very excited to hear your talk today about manganese and redox processes. So thank you so much, Sam, for accepting. We know you are very busy, and it's truly an honor and a privilege to have you here giving this talk to us.
Well, thank you, Tiago. Okay. So that's the first thing I want to say is to thank Tiago. And also, as you mentioned, Lucas made a huge impact on our relationship and really bridged our universities together. And so this continuing collaboration that's brought Hermano into our lab group and become part of our family and in California has been amazing. So I'm super excited to go to Brazil. I've always wanted to go. So great. Hopefully the pandemic will be over soon. So
yeah, let's hope for that.
Thanks so much to everyone at USP for setting this up. And so I will be giving a talk that's hopefully gonna be within the time constraints, and then I'll take any questions afterwards. So thanks for being here. Okay, so I'm gonna go ahead and share my screen and then put it onto full screen. Can you see that? Okay.
That's perfect, Sam.
Okay, great. Yeah. All right, so here we go. So I'm going to tell you a little story about something that I've called a silent killer. And it's because to me and a number of people around the world now, it is an under-recognized contaminant that is globally distributed in groundwaters. And I've purposely named it that so that you can kind of see how this story has started uncovering itself as I've gone through this path of biogeochemical research over my career. So this is kind of like almost like a little thought process that in terms of research for how I've kind of gotten into studying contamination of groundwater. So this is not work at all that was done only by me, of course. There are a huge number of people that contributed, including Michael Schaefer, who is now a professor at New Mexico Tech, and then Scott Fendorf at Stanford, Miranda Aiken, who is a graduate student in our lab, Maitali Ramachandran, who is a postdoc, and Kurt Schwabe, who is a professor of public policy at UCR. So it's a very interdisciplinary group, and I'm happy to share our findings with you. All right, so when we think about water resources in the world, we often have an image when we think about jeopardized water resources, meaning not having the right type of water. And so when we think jeopardized water, we usually think lack of water. So we think quantity of water is very important. And these are images from a few years ago in California. We had the worst drought ever, as you can see on the left is a farmer in Central Valley, and they didn't have enough water, both from rain and groundwater, to have the same crop yield as usual. And on the right is an image from Western Africa that is experiencing more and more intense drought. And so when we think about that jeopardized water resource, if we're biased toward that, we're actually missing a giant part of the equation, which isn't necessarily just quantity, but it's quality. So just because you have a lot of water around doesn't mean that you can use it. And so there is approximately 2.1 billion people and growing, that is estimated by the WHO back in 2017, who don't have access to safe drinking water. And that 2.1 billion people isn't evenly distributed around the globe. It's actually quite concentrated in this square that I've delineated here. 65% of that 2.1 billion people actually live within this square who don't have access to safe drinking water. And their water quantity isn't a problem. So their everyday looks something like this. It's regularly flooded on a seasonal schedule. And so I'm going to tell you a short outline, a timeline of how the water quality story really unveiled itself in that little portion of the globe in Southeast Asia. So before the 1960s, a lot of people drank from surface water. So they'll just have a pond in their backyard, and then they'll go there and their kids will play in it and they'll wash their cows and then they'll drink from it. And so, of course, this common use surface water leads to contamination with surface water pathogens. And so this is an image of cholera, which infected millions of people and ended up causing the world's highest infant mortality rate when that was finally being researched by the WHO. And so to try to mitigate this surface water problem, UNICEF, World Bank, and UNDP went into this region and tried to implement a new source of water. So the idea was, okay, instead of surface water, let's think of something else. How about groundwater? And so that was a great idea. So initially it was, because the sandy aquifer that makes up the subsurface sediment acts as a natural filter. And in that way, it just filters out the cholera before it reaches the surface. So infant mortality rate from the surface water pathogen consumption decreased because the groundwater naturally didn't have those pathogens in it. And it became this great hope, because then as more and more wells were being installed, more people were exhibiting less illnesses from cholera. But the problem was that this groundwater that was being consumed by people was only being tested for pathogens, because that was the major cause of illness. So in the 70s, after not thousands, but hundreds of thousands and now millions of wells that have been installed in this region, people were drinking that water for more than a decade. And eventually by the early 1980s, arsenic poisoning was starting to manifest in people's physical appearances, such as here, where there's pigmentation issues on the surface of their skin, and then also causing systemic cancers. So this, the WHO now recommends a guideline value of 10 parts per billion, or micrograms per liter of arsenic as the threshold amount of arsenic in drinking water. And if we look at where this arsenic comes from, it's interesting, because often people think of arsenic as being a poison, say for getting rid of rats, but really in this giant region, it's a naturally occurring thing in the geology. And so when we look at the Tibetan Plateau, that's really where all these minerals are coming from in the Himalaya. We have these arsenic containing iron sulfur minerals, such as arsenopyrite. And these minerals are weathered from the Himalayan mountains, and they're carried downstream over these giant riverways, including the Brahmaputra, the Mekong, and the Yangtze, and they distribute these minerals in the low-lying lands below through these riverways. And through this transport in these giant rivers, those reduced minerals that contain the arsenic go through oxidative dissolution, and that process has now released arsenic into the groundwater, causing over 200 million people to drink arsenic contaminated water. And so that's a huge problem, right? So for a couple decades now, the question has been, what are the biogeochemical processes that cause arsenic to go from the surface sediments into the groundwater that's poisoning everyone? And so just to dig in a little bit into the biogeochemistry, just on the surface, redox processes and redox conditions are key control to whether or not arsenic is going to be a problem. Under oxidizing conditions, you have arsenic 5, and under reducing conditions, you have arsenic 3. And arsenic 5 is labeled as the less toxic one, whereas arsenic 3 is the more toxic, more mobile form that can spread to farther areas. And so if we look at a soil profile, we know that it's not homogeneous, right? We know that it's actually made of all these little units that we call aggregates. And when we zoom into the aggregates, we can see a different scale of redox that really controls where the arsenic is going to come from and go. And so when we zoom into an aggregate here, we have these larger pores that carry water through advective flow versus these inner pores that carry water through diffusion. And so that transport is much slower. And because of this dual pore domain, the way that oxygen is distributed in our soils is very heterogeneous. So what happens then is you have an oxidizing exterior and then a reducing interior that is dominated by anoxic conditions. And this can cause manganese oxides, which is a very strong naturally occurring oxidant, to accumulate on the surface of a lot of these aggregates and then leaving an anoxic interior. And so we can actually see this when we take aggregates. These ones are from Cambodia on the riverbanks in a region that has a lot of arsenic contamination. You can see that the manganese accumulates on the exterior of these aggregates and around root zones where roots pump in oxygen. And then you have a more iron-dominated matrix otherwise. So these redox conditions, this demonstrates really changes the distribution and dynamics of even manganese. I'm leading you into the story. So what's so special about manganese? In this case, manganese is formed, manganese oxides can go from a dissolved form to a solid form as these minerals that are strong oxidants by microbial catalysis. So microbes take manganese that's dissolved as manganese 2 and oxidize it to form these strong oxidants, manganese oxides, using oxygen as the electron acceptor. And these manganese oxides are very important in the cycling of other contaminants, including arsenic. And so it can take arsenic 3, the more toxic, more mobile form, and oxidize it to arsenic 5, which is less toxic and less mobile because it can absorb onto more minerals in the soil matrix. And it can also do all sorts of other things like oxidizing chromium to the very toxic chromium 6 form, and also making immobile vanadium into the mobile vanadium of vanadium 5. So these manganese oxides play a very important role in controlling contaminant cycles. But what is often not emphasized, so when you read the manganese literature, it's almost always this whole picture that I just painted, that manganese controls arsenic cycling. But as you are oxidizing that arsenic, the manganese itself is being released. And so it is reductively dissolved and you get dissolved manganese being released into the water. Okay, so that's something to keep in mind. So then now we move into the interior of these aggregates where it's highly reducing. And there you can have microbes that are actually going on the opposite side of the reaction and causing reductive dissolution of a lot of existing minerals and then causing the back reduction of arsenic. But in that case, you can have the same thing where you have manganese that is oxidized being reduced through microbial respiration and releasing manganese again into the dissolved form and potentially going into the groundwater. So my point is, even though we've been emphasizing for years the importance of arsenic, all of these processes that are controlling arsenic also have a strong impact on the manganese. And in fact, now we know that manganese contamination is very widespread in places that also have arsenic contamination in the groundwater. And so just to back up and tell you something about how the world views manganese as a contaminant and why it's underappreciated or under-recognized as a problem. In 2004, the guidelines for drinking water quality in the third edition that is released by the World Health Organization set a guideline limit of 400 parts per billion. And then since then, all these studies have come out on the public health impacts and the epidemiological impact of drinking manganese contaminated water, and that it has high or adverse effects on children's intellectual function and learning, particularly for infants and children. And so all these studies just keep coming out and they're still growing. Yet in 2011, the WHO decided to take that guideline value away. And they stated the reason why now they don't have a suggested guideline for manganese is that this health-based value is way above the concentrations of manganese that we normally find in drinking water. Therefore, we don't need it. So in other words, they're saying, oh, 400 ppb of manganese, that doesn't happen. So we just don't need to look at it anymore. But what we did was we went into the literature and found published values of where manganese contamination of groundwater happens. And what we found so far is it's everywhere. So this is just at the start of a list of countries that have published studies that show concentrations of manganese orders of magnitude higher than 400 ppb in groundwater that is then potentially used for drinking. So hopefully I've funneled you now into finding out what that silent killer is, and that if we overemphasize some of the other contaminants, we might overlook some of these less studied contaminants like manganese. So now I'm going to show you in a biogeochemical setup, or like the biogeochemical world, what manganese is affected by, and when you would you might not sorry, now I'm tripping up on this, when you might miss the fact that manganese will be a problem. All right, so two factors really affect manganese. One is seasonality, and that is, you can kind of say that that is essentially redox fluctuations, or causes redox fluctuations in the subsurface where your groundwater is. And then the other aspect is depth stratification of your contaminants. So I'm going to start with how seasonality and redox fluctuations will manifest in different dynamics for different contaminants like arsenic versus manganese. So if you remember, these Himalayan arsenic containing minerals are distributed all over these different low-lying valleys, and these basins include the Yangtze River Basin in China. So our research crew went there with the Fendorf group from Stanford, and we chose this field site in the Jinhan Plain that is just west of Wuhan, a giant, which is a very famous city now, a giant city in the middle of China. And it's an inland river basin that is made of alluvial sediments and has a subtropical monsoon climate. So the landscape looks a lot like the picture I showed you at the beginning of the talk, where seasonally it's very, very wet. And it's dominated by aquaculture there. And that's an important point that I'll bring up in a little bit. So we zoom into our field site here, and what brought us to that field site is because of the incidences of very, very high concentrations of arsenic in the groundwater that people were tapping into. So this is a very rural region where instead of having a public system of water treatment that feeds into houses, people are tapping straight into the groundwater in the aquifer locally and then drinking that raw groundwater. And so whatever the geology is giving them, that's the contamination that they're also consuming. And so you can see in some of these areas, the size of the circle indicates the concentration of arsenic in their groundwater. And in this region, there are many locations with arsenic much greater than 400 ppb. So more than 2 orders of magnitude greater than one order of magnitude greater than the concentration that's suggested by WHO. So there was a matrix of 3 sets of wells that were dug in this region so that we could find out what is causing this arsenic contamination and what seasonality is doing to these concentrations of arsenic in the groundwater. And the maximum concentration within this well setup was 1,205 ppb. So just to remind you, the limit is 10. So we're very, very high here. And then the average is even 6 times greater than the WHO at 65 parts per billion. So this field site was bracketed by a surface water body as a river on the northern side of the village. And that's important because that river had impacts on the subsurface water chemistry because of recharge and discharge. So I'll talk about that quickly here. So we can see that over a year, as shown on the x-axis, we have time, and on the y-axis, we have water level of the surface water in that river. And you can see it goes up and down seasonally because of the monsoonal climate. So during the summers, when it's really wet in the monsoonal climate, we get very high water levels in that river, and then it dips in the winter when there's less rain, and so on and so forth. And we can see the seasonality in the surface water level. And then when we look at the groundwater level, of course, we see a very muted seasonal effect compared to the surface water. So what we can do is take the difference between these two, and it will tell us the direction of water flow. And the way that works is that during the monsoon season, which is during the summer, you have very high water levels for your surface water in your river and relatively lower surface or level of groundwater. And when this happens, you have groundwater recharge, which means that your surface water is feeding into the aquifer. Whereas during the dry season in the in the winter, you have the opposite, which is that your groundwater level then is actually higher than your river. And so that actually causes groundwater discharge. Okay, so you can see how the water sources communicate and may affect each other seasonally. So what we've shown here is, again, on the x-axis, we have time, and on the y-axis, we have the groundwater level, but with a difference from the surface water. So the surface water level minus the groundwater level, which means that if the value is positive, we're having groundwater recharge. And if the value is negative, we're having discharge. And so we can take those numbers and we can see the seasonality of when we have groundwater recharge versus groundwater discharge. Now we can look at what the chemistry looks like. And what we see is the arsenic looks like this. So not surprisingly, we see this seasonality in here I've just displayed at the 25 meters, but we see this in all of the wells, that there is this up and down that follows the seasonality of the water levels and demonstrates recharge and discharge effects. So the decrease over 3 months can happen from November to May and it can change the arsenic concentrations by 10 times during recharge, versus when it's discharging, we can have a 10 times increase over only 2 months. And so what this is showing you for just even looking at arsenic is that testing water in wells for their water quality based on arsenic concentration is really dependent on when you do it, right? So that is one of the major messages that we got here. So when we look at other chemical compounds that are in the water, we also see this seasonality in sulfide, iron 2, and different forms of arsenic. And we see that we have recharge periods and then discharge periods really following what we're seeing in the arsenic. And in the end, what this does is as I mentioned, depending on when you're testing these wells, if you're testing this set of wells during a time where the arsenic concentrations are low, then you can have up to a 15% of your wells being labeled with false negatives, meaning that you're saying these wells are safe when in fact, most of the rest of the year, they're not. Okay, so what about the manganese? This is what the manganese looks like. So unlike arsenic, the manganese is constant, which is fascinating because of course we think of manganese as a redox active element. But as you can see, there's a lot of other geochemical parameters that need to be taken into consideration aside from your typical oxygen and nitrate elements that affect the redox. And so there's a lot of redox limitations to when you get rid of manganese from solution phase. So just to show you how the manganese concentrations look, down here is that WHO guideline value that was set back in 2004. So no matter which well you were looking at, the manganese concentrations were way above it and were constant throughout the year. And is another huge problem that isn't being looked at. So what we think is happening is that we're transitioning from oxidizing to reducing conditions when it's discharging from the groundwater into surface water. And what's happening there, if we zoom into that matrix, is that you don't have oxygen. And so you're transitioning from a more oxidized environment that's shown here, where you might have iron hydroxides and then some manganese oxides that have arsenic that's oxidized as arsenic 5 adsorbed to these solids. And we can confirm that using X-ray absorption spectroscopy. And when the oxygen goes away seasonally, all these minerals and metals that are redox active become reduced. And so we have reductive dissolution of the iron and manganese minerals, and that causes arsenic to be released, including the manganese, and that can then go into your well that we're testing. Whereas during the recharge, when the surface water is high, what we're doing is then flipping that. We're doing the opposite, where we're going from reducing conditions to oxidizing conditions. So what we had in the previous slide in the previous season, we had all these reduced forms of the metals, and we now have these oxidants that are coming into the environment, partially because it's dominated by aquaculture. So aquaculture, they actually pump oxygen into the surface water here. And so when you have the recharge happening, it's actually leaking all this nitrate that's from the fish poop, and then all this oxygen from the aeration goes into the groundwater. And that causes any of the iron that's left over from the reducing season to precipitate as iron oxides. And so then that can become the sorbent for the arsenic that might be floating around. And so that causes that decrease in the arsenic that we see during the recharge season. But when we look at the manganese, what's happening there is manganese oxidation by oxygen abiotically is extremely slow. So it's not like iron. And instead, it can stay in solution for a very long time if there is no microbial catalysis to form manganese oxides. And so this is a very, very slow process. So unless you have enough microbes around that are catalyzing the formation of these oxides, you end up building up the manganese concentrations over time. So this is our hypothesis of the likely reason why the manganese concentrations are very constant and remain high. So the second aspect that we see that's affecting our kind of like the way that we're handling the manganese versus the arsenic is depth stratification. So in all of these places that have the arsenic contamination, we went back into these fields and we dug through the data and found all of the manganese contamination data of the groundwater also. And the four regions that we looked at were China, Bangladesh, Cambodia, and in the US. And we took all this data and we looked at their distribution of arsenic versus manganese contamination with depth. So this is what their subsurface profiles look like. Most of them are clays overlying a sand interbedded layer below with clay. So some of them have these clay lenses and some have very sandy aquifers that are below a clay layer and so forth. And all of them here on these three show a more dense surface horizon and much sandier subsurface, of course, because that's the aquifer that people are drawing groundwater from. And so this is the distribution of number of wells as a function of depth. So on the y-axis, we have depth, and on the x-axis is the number of wells. And over this, I'm going to show you the arsenic versus manganese contamination. So when we look at arsenic contamination, these lines show you the percentage or fraction of wells that are contaminated with arsenic as you go down. So in China, the surface wells, where a majority of them are placed, have a relatively low fraction of them that are contaminated with arsenic. But as you go down with depth, you have more and more of them, and so on and so forth. And you can see the shape of this curve is consistent across all of these regions. And now when we overlay this with the fraction of wells that are contaminated with manganese, it looks kind of like the opposite. We have more fraction of wells contaminated with manganese, and then there is always a crossover point, oops, that we see at a certain depth where more of the wells are contaminated by arsenic, and above that, more of the wells are contaminated by manganese, shown in red. And so what I'm showing you in this slide is the hashed area here that is shown in the white line is the number of wells at every depth that would be an underestimation of contaminated wells if we don't take manganese into account. So it's just showing you that a lot of the surface wells or the shallower wells are being labeled as safe because we're only looking at arsenic and not necessarily looking at manganese. And what we show also here is that it's statistically significant every time in each of these regions where we're taking thousands and thousands of data points, that there is always a significantly shallower maximum number of wells that are contaminated with manganese. So this relationship isn't just qualitatively us looking at these graphs. We can actually demonstrate that they're always significantly shallower wells that are manganese contaminated. So when we look at the redox, we see a similar trend as I showed you in detail for China, is that arsenic shows a very strong redox correlation, whereas manganese is very poorly correlated with redox potential and dissolved oxygen. And it's likely that it's because manganese oxidation is kinetically limited. So what we think these results can show you is that we have implications for what the water can be used for. So these deeper waters, just don't drink it because it's arsenic contaminated. Whereas if you're tapping into shallower wells up here that are potentially both arsenic and high manganese, you could use that for irrigation because a lot of plants and crops are not as affected by manganese uptake. So one of our suggestions then is you might know the depth of your well, and that might have some input on which contaminant is dominated and therefore might tell you what's the best use. So if the deeper water is contaminated with arsenic, then rainwater is then ultimately probably your best next option. So what this has implications for also is that we have these papers that have been published that show you well level indicators. And one of these ways of telling whether or not your well is safe, this was published in 2012, was to just look at the color of your well skirt. So wells are drilled here, and then a perimeter is usually placed around the well that people stand in, and then you pump the water out, and then you take that water to your house and you drink it. And so some of the water that's left over will then oxidize over time and precipitate different colors. And so what this paper was demonstrating is that if your well showed black precipitates, then 73% of the time you are arsenic free. Your water is then labeled as safe, versus when your well pumps out water that looks red because of the iron that is in your well water, then that tends to have a higher chance of having arsenic contamination. So their suggestion was, if it's red, don't drink it. If it's black, you can drink it. But what I'm showing you is when it's black, what that means is it's full of manganese. And so this is just a demonstration of the fact that we've been very arsenic oriented. And since more and more of these public health studies are just demonstrating that manganese can have a very high impact, particularly on children, we might want to revisit these types of suggestions. So this is not only studies that are relevant in those regions that I talked about for manganese, but there is a very, very local problem for us within the US. And part of it is because manganese is a secondary contaminant that is unregulated, which means that any guideline values are not enforced. And so they're not federally enforceable, even though some states have some guidelines. There are still not any health-based guidelines that are being enforced throughout the country. And so we can do the same type of map where we see that states that have water utilities reporting higher than 100 parts per billion of manganese is pretty widespread, including here in California. And a statistic from the California Division of Water shows that 30% of California drinking sources contain manganese at 10 times the secondary limit, which is 50 ppb. So this is 500 ppb of manganese. And so who's drinking this manganese contaminated water when we think about Californians? So there's essentially two different categories that you fall into when you're drinking water. You have public water, which is monitored and treated regularly. And then you have private water, which is called also domestic water, that is not monitored and not treated. And these private wells tend to be shallower wells, whereas the public wells, because it needs to extract more water, tends to be deeper. And as you remember from our last section, shallower wells tend to be dominated by manganese contamination. And so you have higher probability of people drinking from private wells who tend to live in more rural areas and tend to be poorer, are the ones that are going to have a higher exposure rate to manganese. And then they have a much higher chance of having poisoning because manganese is unregulated and unmonitored. So about 2 million Californians use private wells as their primary drinking source. So these 2 million people are likely open to this manganese exposure that's not being looked at. And 31 million obtain a portion of their drinking water from groundwater. So we see this actually because it's not enforced, there's less money going into looking at manganese, but communities still complain and they still see the effects of it. So we actually see where manganese is a problem in the popular press more than we're seeing it in the research right now. So we have these news articles that come out all over the state and the country that show people in a lot of these more rural, isolated communities having water that looks like this coming out of their tap. Sometimes it's from the infrastructure of their pipes, and sometimes it's because they're tapping into the shallow groundwater. And a lot of these communities have to then purchase alternate water, and they're buying these bottled water packets, which can cost up to $130 a month, which is a lot in the US. And again, these communities tend to already be the poorer communities. So to top it off, just to get clean drinking water that's manganese free, they're having to pay for additional water. So one of the main paths of where our group's research is going now is we're trying to identify what we call vulnerable communities, which communities are disproportionately exposed to high manganese and also high arsenic concentrations. And this causes what's called health disparities. So that means that some regions, depending on certain aspects of the demographics, including their income, are being impacted more and in a worse way than other communities just because of where they live and that they don't have as many resources to have safe drinking water. And so we're taking a lot of state data to look at the groundwater chemistry along with their drinking water sources. So are they drinking public versus private water? And then taking infant vital stats. So what is the infant mortality rate, and what is their birth weight, and what was the condition of the mom when she was giving birth, etc. And then also looking at child cognitive health. So intellectual growth and attention span in schools. So taking all of that data, we're trying to overlay them together with the groundwater chemistry, and then looking at whether or not there are certain communities throughout California that are being exposed to manganese and arsenic and not getting the treatment that they should, both for the human health and also for the water quality. And this is a huge problem across the world, but it's also a huge problem just locally within our own state. And so that's why we've kind of started looking both near and far for what the impact of these contaminants are on communities. And with that, I am done. I am right on time. And I'd like to thank a huge list of people, but I actually would like to highlight the daycare that provided a lot of the childcare that I've always needed. When I was in Northern California, I think especially right now during the pandemic, we need to acknowledge people who have made a huge difference in our lives to make science move forward. And that includes people who help us with our children. So I would like to thank her with raising my children, and then a lot of collaborators throughout the world, including in China and Cambodia, and a lot of fantastic people at UCR. And that's it. Thank you.
Great, Sam. Thank you so much for your excellent presentation. We have a lot of questions. I have a couple of them, but I will start with the questions sent from the people that are attending your talk.
Okay.
So the first one, as you must expect, is from Hermano.
Great.
So I'm going to read it. I have another screen here. So, Hermano, congratulations you for your amazing presentation. And he asks, manganese is often overlooked contaminant due to the large amount of this element in the environment. He asks if you think the threshold should consider not only the concentration, but also the biogeochemical conditions of soil, water, and geology, and also indirect effects of as release of toxic metals as arsenic. I believe he's referring to the threshold limits of 0.4 ppbs. Is that right? Mm-hmm. If you should also add more information to that threshold to be safer.
Yeah, so that's a great question. And that's kind of where we're funneling to when I was talking about the kind of overall impact on communities. And so that's kind of, I think Hermano mentioned biogeochemical kind of like conditions, right? Mm-hmm. But I think on top of that, we're also taking human conditions into it to think about what the cumulative impact of all of these contaminants are. And he's asking a very important long time question that we've had, which is we have these guidelines on individual metals, but we now have some of the computational technology to start thinking more about what is the cumulative or compounding effect of multi-contaminants, right? Mm-hmm. Which I've just introduced too. And of course, now we have PFAS and PFAS and all these other, and chromium. And so I'm only touching the tip of the iceberg on that topic. But yeah, I totally agree that there needs to be a more systems way of considering water quality. And the trick is, how do we present that, and how do we kind of quantify it? And then how do we communicate it so that it's not alarming to people? So that is definitely like the direction that we need to go, but also taking into account who has the resources to treat their water, right? So we tell them that it's contaminated, but it's only impacting the people who can't treat it. So that's also a consideration.
Yeah, great. So I have another one, Sam, from Amanda. So Amanda is thanking you for the excellent talk, and she asked, how did you differentiate the source of arsenic and manganese contamination? And what she means is, how did you know that the source of manganese was not industries or mining activity in South Asia?
Okay.
In South Asia.
Yeah. So part of it is just that some of the subsurface data is coming from extremely deep. So I didn't dwell on that data too much, but we're like, in Bangladesh, it's like hundreds of meters, right? They dig very, very, very deep wells. But so in that case, if it were anthropogenic, it would have had to go kind of through the well. But also just the landscape there in general. A lot of these, and I didn't go into it, it's a good question, that the land use there is not industrial in almost all of Cambodia, for example. So there's just not actually a industrial manganese source for a lot of those. And also the other way we can tell is if we were to place it on a map, the manganese distribution is very widespread and it's not correlated with where the industries are.
Okay.
And it's a function of the geology.
Great, Sam. There is another one from Diego Barcelos. Could you tell us more about the slow oxidation of manganese in the systems you worked? And he mentioned a recent paper from Aaron Thompson and Chunmei Cheng in Geoquímica and Cosmoquímica Acta, showing that the presence of soil organic matter decreased the rate of iron oxidation, whereas the well-crystalline forms of iron increase the rate of iron oxidation. How that compares with manganese 2 oxidation?
Oh, that's a great question. So, of course, first of all, the dynamics of how secondary transformations happen with iron is initiated by abiotic oxidation with oxygen. Is, of course, the kinetics there are totally different between manganese and iron oxidation. And the second part is because of the kinetic limitation of manganese oxidation, essentially requiring oxygen, even though there's one paper that just came out that shows that there is an abiotic process that is being introduced. But nevertheless, without the microbes, that process is so slow that it's going to, just in the timeline form, is going to look very different. Second thing is concentration of manganese is relatively low compared to iron. So going back to the question, if you have organic matter involved with the oxidation of manganese, I think that you could have some similar impact in that you have ligands that can associate, especially with manganese 3, that stabilize the manganese 3 form, for it contributing to the different crystallinities of the resulting manganese. If we don't take kinetics into account, it does have a similar impact in that you have some manganese 2 floating around, it resorbs to your manganese oxide. It will contribute to decreasing the average manganese oxidation state within the oxide, which then makes the oxidative power decrease and you can have surface passivation. But in terms of it like undergoing secondary transformation, it does also do that, where there's some past papers, including some of ours, that show this transformation to more manganese 3 dominated forms, and then like more and more manganese 2 with on the surface. So I don't know if that answers the question.
Yeah.
It's essentially like, yes, those processes do happen also, But because it's a lower concentration and because you have kinetic limitations, the impact of those transformations are going to be quite different on the soil environment.
Great.
Sorry,
So
I don't know if that's a great answer. But
I will continue reading the questions, but I will make one of mine
sure.
questions. So Sam, you showed that the sampling wells for contamination, arsenic or manganese, is a very difficult approach to assess the real risk for contamination, right? So is there any overall guideline for WHO parameters that is not just, I mean, you showed huge seasonal oscillations and depth oscillations. I mean, how the WHO approaches these new data or the monitoring is, I mean, you can rely on it, right? So what is the way out?
That's a great, so one of your first questions that I should know and I don't is what are any guidelines that WHO have on sampling? I don't actually know that, and that's a great question. And then the second one is, even if they have a guideline, it's not, internationally enforced. And so what we saw in China is that even when you have government resources, including people coming out to help test and monitor the wells, resources are limited. When you send a person out to sample and then test the water, but you're showing this even monthly, variation in the concentration, are you going to spend that money on every single well to test it all year long? So we're showing this, but like you're pointing out, what is the practical resolution? And honestly, I think that it's a technology question. So there is a collaborator at Caltech that this is a technology that's developed in a lot of different venues, including for health monitoring. They have these teeny tiny sensors now that I think they can hook it onto anything. A lot of them are for people to wear, and usually then they can see what the environmental exposures of people are over time. It collects concentrations and sends data through Wi-Fi for what people are exposed to. And the technology that he's trying to develop now is pretty much a tiny device that you can hook onto something like a well, where the well outputs the water, or on somebody's tap, and it can take data on what the elemental composition is. And so I think some sort of, technology like that is really needed to do this like high resolution monitoring. I don't think with the current way that we're monitoring water is possible. I
mean, people are in the dark, right?
Yeah, exactly.
The current situation, yeah. So we have one from Gabriel Nuto. Is there any process, mainly based on biogeochemical approach, that could revert or treat this high arsenic and manganese concentration in drinking water?
Yeah, so they're already in public treatment systems. There are plenty of options for removing arsenic. Actually, in Europe, manganese oxidizing bacteria are sometimes put into public treatment to get rid of arsenic, because it can oxidize arsenic, and then you can remove it by adsorption onto some sort of surface, including iron oxides. Green sand filters are an option for manganese removal. So one of the things that we actually just wrote an opinion piece that is stating exactly that. The treatment for manganese contaminated water is thoroughly known and economically feasible. So point of use treatment with just a green sand filter, anything that just quickly oxidizes it, and initiates the formation of manganese oxides, you're kind of like then are growing your own filter, because once you have the manganese oxides precipitated, you add more manganese to it, it'll precipitate out more, and then you just filter it, and then it's clean. It's just a matter of monitoring and then finding which communities need the point of use.
That's great. Let's see, we have one from Felipe Hipólito. Are there any actions being taken in mass or not to turn the water safe or to make the water safer to drink or to use in agriculture? Actions like those taken by ONU, installing the wells now to treat or filter the groundwater. I don't know if you got it, Sam. I'll repeat the first one. Okay. If there is some action to turn the water, I believe you already answered that, right?
And is he talking about it specifically in an agricultural
For
side?
Agriculture and drinking water.
Yeah. So for agriculture, I think usually not for manganese. Yeah. So, and the difference with agricultural use is that plant uptake of these metals is not actually as much of a problem if you think of the uptake and then the consumption being a source of manganese and arsenic for people versus actually directly consuming the arsenic contaminated water. So if you were to pick one, you would rather eat crops that have those metals because the plant itself tends to be a filter for some of the concentration.
Oh, that's a great answer, we have two that are very similar regarding the bioremediation strategy. If you know, it would be a viable strategy for reducing manganese contamination. And how common are manganese oxidizing bacteria in soils?
Oh, that's a great question. I like that question because I started as a microbiologist, and the first project I had was culturing manganese oxidizing bacteria. And so they're everywhere. Literally, I could take soil from my daughter's playground and just put it on a manganese 2 containing plate, and you'll see these colonies grow that have black dots on them because they're oxidizing the manganese and forming the oxides. So they're super common. They're very widespread. They're poorly understood because people still don't totally know why they do what they do. And that's what I was mentioning. There's one paper that just came out that implies that they might be doing it for energy, and that hasn't been known. So they're very mysterious microbes, and I like them a lot. And it's not just bacteria, it's fungi also do a majority of the catalysis. So the answer is they're everywhere. And then for bioremediation, for manganese, you could just use them. So we use the Leptothrix for a manganese oxidizing bacteria that doesn't form spores in water treatment because they form the manganese oxides. Those manganese oxides themselves catalyze manganese oxidation because they're autocatalytic. So as long as you have those as a source, you can do arsenic and manganese at the same time.
That's great. We have another from Diego. Broadly thinking, in the tropics, scientists often think that some soils are constantly aerated, such as oxysoils in Brazil. I have been thinking of the redox dynamics within microaggregates in these soils involving manganese and iron. For instance, the agricultural inputs of fertilizers with trace elements and pesticides may increase contamination. What do you think for the potential to look closely at the microaggregates, redox dynamics, and metal contamination in the Brazilian oxide soils?
I think he's just trying to make me really happy by asking that question. Obviously, I think that's an excellent question. I think that it takes into account the inherent heterogeneity of our soils that are it's becoming more and more of a phenomenon that people are recognizing, that you have these anaerobic microsites that are due to the aggregated nature of most soils. So it's another, similar to the question that Hermano asked, in that it's moving toward a systems way of thinking of contaminants, right? And really taking into account not just the chemistry, but the physical structure and the integration of the biology with how it changes the contamination dynamics. And so the answer is yes, which is that I think it's a great idea to kind of to dig into it more, to look at intact soils and you know, where the contaminant distribution actually is, and knowing that requires you to know what the redox interfaces are. And then after that, you take into the flow, right? So then where they are, if it's diffusion limited versus advective flow, that will tell you something more about how fast those contaminants will not only cycle, but be transported into the subsurface. And so that whole systems way of thinking is rigorous and resource intensive, but important, you know, to really see where things are going.
That's great. We have a few more. Carla Silveira, is it possible to see the seasonal differences in the concentration of these metals in the soils? And I believe there is a second part. I was wondering if manganese contamination have any effect on wildlife, and if you would have some information concerning this.
Oh, okay. So for the first question, is the question, what's the manganese distribution in the solid matrix?
I think so. If there is a seasonal difference.
Yeah. So it would be like the slide that I showed with the aggregate that we collected from the riverbank in the Mekong. You can see that manganese distributes itself in the oxic, you know, portions of the soil because of the need for O2 to oxidize the manganese. And then what I showed you there is what the distribution in the soil matrix looks like when it's under dry conditions, right? And then of course, when it's wetted up, that manganese that was on the exterior of the aggregate is going to reductively dissolve. So you do see that dynamic also. So it's almost like the reverse of what you see in the aqueous phase. When it's high in the solid, of course, then it becomes dissolved and high in the aqueous phase. For the second question, the only thing I know about manganese impacts on animals is from Hermano's research with the fish. So I know that fish can accumulate a certain portion of it if they're even just living in manganese contaminated water. And then I also know that it's quite toxic to animals like horses. So if you raise horses in a swampy area, you have to be careful of manganese reduction.
That's great. We have another one from Gabriel Nuto. Another question is, isn't time to start thinking integrative? That's very similar to Hermano and what we were talking about. And he means geological, pedological, hydrological indexes to express water quality.
That's a great idea. So that's moving toward a quantitative,
an integrative index for water quality regarding soil, hydrology, geology.
Yeah. I think that's a great idea. And similarly, I mentioned that our lab more and more is moving toward the social, like societal impacts of these water resources and the kind of disparity in their distribution. There are different vulnerability indices similar to that are being developed also. The trick with those is you just have to be extremely clear and transparent with what the calculation is that's going into that one number, because anything that we create that's like that, an index, has so much data embedded within it that if we're, you know, weighting arsenic greater than manganese, we need to be clear about why we're doing that and when what the buffer to that value might be, so the index should probably have a value and then a plus or minus always to go with it.
Yeah, I think that the greatest challenge is how fast things change from a biogeochemical point of view, right? Because if you sample a well, after a heavy rain and before that,
it's gonna be totally
probably have two different worlds
in
terms of manganese and arsenic. That's when you are monitoring wells, you are like taking a snapshot from one moment, and it may mean nothing. So your answer on the tech guy, the micro tiny sensors, probably
that would be amazing.
Yeah. I don't know if we have more questions. I will ask Raissa to give me some directions here. If they don't have any more questions, I have one last one for you, Sam. We have one more here. Hermano is typing. Oh, from Lucas.
Cool.
Yes, finally. Hey, Sam. Good meeting you again.
Yay.
Do you think clay minerals, of course, he would mention clay minerals. Do you think clay minerals also play an important role on arsenic absorption in these environments?
Oh, interesting. So unlike Lucas, I don't think about clay minerals as much. Mostly because we've worked in these, monsoonal tropical regions that are really dominated by iron oxides, right? So, yeah, for sure, there's a lot of studies that look at manganese and arsenic interaction with phyllosilicates. I am not an expert at that at all, actually. So for sure, they play a role. How much of a role, at least in the regions that we were studying, it's definitely restricted to kind of like the surface layer, like I showed you in the profile, that is more dominated by clays that are actually expandable phyllosilicates. You can see during the dry season, the cracks are like this big. And then in the winter, of course, it's just covered and totally sealed up. So in that way, it affects also the communication of surface water with the aquifer seasonally, because the percentage of smectites is just so high that you get a lot of preferential flow at the very beginning of the wet season when the cracks are still there. In terms of like sorption, I think that still the iron oxides are really dominating as the capacity there.
Okay. So I don't know if we have more time. Raissa will tell me soon in the chat. So Sam, do you think that in US, you mentioned at least in California, a lot of wells have 500 ppb, is that right? A large number of wells.
Yes.
Okay. Yeah. So, and as you said, those wells are mostly located in rural areas. Is that right?
Yes.
So is there any cases of human severe contamination and problems? Is this starting to appear in the US with manganese?
So there are a couple studies that have looked at communities on the East Coast, actually, in North Carolina and in Massachusetts, that showed impacts on children. And they and from my memory, there is a range of health metrics that they used to look at their impact. So as a public health study, there has only been a couple that in the US. The largest number of those studies have been done in Bangladesh and then a lot in Canada. In California, in California, there have been none. And that is essentially what our project is. That's being funded by NIH, is we're saying that there's plenty of people consuming manganese and we have no idea what the health impact is. And we have precedent to think that it's going to have impacts because of these other studies that are done in other countries with people drinking similar concentrations. And the other impetus is that in countries like Bangladesh, manganese is a primary contaminant. So where they have more knowledge, it's becoming more aware, and people are more aware of it needing enforcement on the federal level. In the case of the US, it's just a matter of there not being enough information. So to answer your question, we don't know. That study needs to be done. Yeah.
And you mentioned problem with kids, right? With children.
Yeah.
It's what kind of health issues?
So
it's a cognitive problem. I think I saw something in your slide.
Yeah. So it crosses the blood-brain barrier. So it has a lot of neurotoxic effects on a molecular level. I have a graduate student, Ben, who knows this stuff in and out. I'm not as familiar with it, but the effects that have been documented is just that their intellectual function is statistically decreased. There's a number of metrics that people have used for that, for like attention span and attendance. And some of those metrics are debated because there's so many socioeconomic, embedded
that will impact
problems with it. But just zooming in on a molecular level, the manganese studies show very clear effects on neurotoxic. So just looking at its effects in the brain and that it gets to the brain, that's been proven over and over. And it also, in adults, manifests like Parkinson's. So it will cause Parkinson's-like impacts. And so that is already very thoroughly studied.
Great. So, Sam, I believe we don't have more questions.
Okay.
So I would like, again, to thank you so much for your time and availability to be here and to give this great talk. It's a privilege for us. I hope we can have you back here with us in Brazil, in Piracicaba.
Yeah,
right?
That's great. I'm so excited. And it was so great to get comments and questions from Hermano and Diego and Lucas. That's so awesome.
Yeah.
They're fantastic.
So
group.
Thank you so much, Sam, again.
Thank
you. We'll probably be together here in Brazil very soon, hopefully, and carry on with our collaborations with those guys.
That'd be excellent. I'm really looking forward to it.
Ok. Thank you so much. Boa noite a todos. A gente encerra agora com a professora Samantha. E a gente se vê numa próxima. Valeu.
Thank you.